So, very good morning, everyone. Welcome to this first meeting biochemistry. So, today we embark on an exciting journey to understand fundamental aspects or concepts that forms the basis of life and biochemical processes. So, this unit will cover a broad range of topics starting from origin of life to essential measurements and calculations in biochemistry. So, let's dive right in. So, here we have biochemistry introduction score. Biochemistry is basically a branch of science that explores the chemical processes and is related to mechanism. It basically is a laboratory-based technique or discipline that combines both biology and chemistry. Biochemistry studies structures, functions, and interactions of biological macromolecules, such as proteins, nucleic acid, carbohydrates, and lipids, which provides the structure of cells and perform many functions of life. So, thorough understanding of these molecular mechanisms in biochemistry helps us to elucidate the complexities of biological systems that plays a critical role in advancement in health sciences, medicine, agriculture, and environmental science. So, here we have applications of biochemistry, as mentioned, medicine, agriculture, environmental sciences, biotechnology, and many more. And medicine, when we talk of, it deals with understanding of biochemical processes that is very critical in developing new therapies and drugs. Similarly, in agriculture, it helps to improve proteins. In environmental sciences, it deals with the study of population and effects on ecosystem. And in biotechnology, it deals to understand several techniques, genetic engineering, and synthetic biology. So, next, talking of importance of understanding these life processes in biochemistry. So first we have to get the fundamental knowledge. So biochemistry basically is a detailed understanding of molecular mechanisms that underlie various biological functions. This fundamental knowledge is essential for comprehending how living organisms operate at cellular and molecular level. Now talking of medical advancements of biochemistry, so by understanding the molecular basis of disease, a biochemist can contribute to innovative therapies and improve healthcare solutions. Talking of disease diagnosis and treatment, so a clinical biochemist or clinical biochemistry plays a pivotal role in diagnosing diseases and monitoring patients' health, including analysis like biological markers or biochemical markers present in body fluids, helps in early detection of diseases and effective management. Next, we have biotechnology and genetic engineering. So here, as mentioned, biochemistry is the backbone of biotechnology that enables the manipulation of genetic material for applications in medicine, agriculture, and industry. Agricultural improvements, nothing but here, biochemistry aids in enhancing the properties, developing pest-resistant plants, improving nutrition value, or sustainable agriculture food security. Environmental conservation, on the other hand, helps in understanding the impact of pollutants on the ecosystem and in developing strategies for environmental protection and remediation. Next, we have nutrition and metabolism. So, studying biochemistry allows for deeper understanding how nutrients are metabolized and utilized by our body, leading to a better dietary recommendation. So, these all will be discussed in this unit further. Next, we have the fascinating story of origin of life. So here we'll discuss Miller's experiment, which was pioneering demonstrating how organic molecules could form under prebiotic conditions. Now, here in the image, you could be able to see the conditions on early Earth, which was vastly different from now Earth or Earth we experience today. So around 4.5 billion years ago, the early planet underwent a period called perihelion, which was characterized by volatility. So during this time, Earth was bombarded by asteroids, comets, leading to high temperatures and formation of molten surface. So the atmosphere basically that time comprised of various gases such as methane, ammonia, water vapor, carbon dioxide, nitrogen, and hydrogen, and very little or no amount of oxygen. So as Earth cooled, the water vapor condensed to form oceans, setting the stage for chemical processes that might lead to origin of life. So the early atmosphere and oceans, which were rich in chemical compounds, along with volcanic activity, lightning, ultraviolet rays, radiation from sun, provided the energy or necessity to drive the chemical reaction that would lead to origin of life. So here we have some theories on the origin of life, which is like primordial soup theory, hydrothermal vent hypothesis, panspermia hypothesis, and Army world hypothesis. So talking of primordial soup theory, proposed by Alexander Baring Davis LJ in 1920s, this theory basically suggested that life began in a soup of organic molecules, possibly in the ocean. Now, they hypothesized that simple molecules were combined under the influence of external energies could lead to formation of more complex or organic compounds. Next, they proposed, some other scientists proposed hydrothermal vent hypothesis, which suggested that life may have originated from hydrothermal vents on ocean floor. And these vents release hot mineral-rich water, creating a unique environment that would foster the synthesis of organic molecules. Next, we have panspermia hypothesis. This was another great hypothesis that says life did not originate on Earth, but instead it was brought here from elsewhere in the universe, possibly by comets, asteroids, or meteoroids. Now, this theory does not address how life originated, but rather it said how it spread across the cosmos. So, next theory, which most of us believe, is RNA world hypothesis. So, this hypothesis basically states that RNA was the first genetic material arise before DNA and proteins, and it is capable of storing genetic information and catalyzing chemical reactions, making it a likely a candidate for the first self-replicating molecules. So, all these theories of origin of life would be discussed in this unit in detail. So, next, as I mentioned, Miller's experiment was vital to support the first theory. So here, in this experiment, the Stanley Millers in 1953 conducted an experiment under the supervision of Harold Urey at University of Chicago. So this experiment basically provided the first experimental evidence that organic compounds that are essential for life could be synthesized from inorganic precursors under the conditions that are thought to be prevailing on those time of early Earth. Now, the synthesis of amino acids in Miller's experiment provided crucial insights into how life's essential molecules could be formed from simpler inorganic compounds. So here, as you can see in the diagram, Miller designed an apparatus that simulated hypothesized conditions of early Earth's atmosphere and ocean. So this setup basically included a closed system that comprised of gaseous mixture, where a glass was filled with mixture of gases that is methane, ammonia, hydrogen, and water vapor that represented the primitive atmosphere. Next, we have continuous electrical spark. So here, electrodes were used to generate electrical sparks within the gaseous mixture, simulating a lightning and a common energy source on early Earth. Now, boiling water, a separate flask of boiling water produced the water vapors that simulated the early ocean and maintained the continuous cycle of water. Next, we have a cooling condenser. After circulating through the gaseous mixture and electrical spark, the vapor cooled and condensed, mimicking the rain, which returned to the boiling flask, maintaining a cycle. So the experiment ran about a week, during which the gaseous mixture was continuously subject to the electrical sparks, and this setup allowed for the repeated exposure to energy, driving reactions among the gases. So at the end of the experiment, Miller analyzed that the content of water and that was found, and several other organic compounds have formed. Notably, that is amino acids, which are the building blocks of protein and essential components of living organisms. So among the amino acids produced that time was glycine, alanine, and aspartic acid, which we know today. So this experiment we will be studying in detail in the classes in this unit. So next here, we will explore different types of organisms, its classification. So basically, classification of organisms could help biologists to understand the relationship among different forms of life and to organize the vast diversity on life on Earth. The classification is very necessary. Classification system, sometimes also known as taxonomy, is basically based on division or classification of groups or organisms based on their shared characteristics and evolutionary history. So here is a concise overview of main classifications. The first classification starts from domain. Domain is the highest rank in biological classification. So three domains are majorly known here: is archaea, bacteria, and eukarya. Further, this domain was further divided into four kingdoms, that is Protista, fungi, plants, and animalia. Within each kingdom, the organisms were further divided into phyla based on major structural and functional differences. Say, for example, in animal kingdom, the phylum Chordata includes animals with the like fish, birds, and mammals. Now, phylum is again divided into classes, that is, we can say based on different classes. Say, for example, class Mammalia includes mammals characterized by the presence of mammary glands in there. Next, classes could be further divided into orders. Within class Mammalia, the order Carnivora would include cats, lambs, and tigers. Now, orders are further subdivided or broken down into families, and families are broken down into genera or genus. The genus Panthera could include larger cats, like lions, tigers, etc. So, the most specific level of classification is the species. A species is a group of individuals that can interbreed and produce fertile offspring. For example, Homo sapiens is the species name for humans. So, similarly, we would be classifying the organisms, and we would be reading about the classifications in details in this unit. And next, we will explore on the types of organisms, prokaryotes, eukaryotes, the distinction between unicellular and multicellular organisms. So, basically, understanding these basic classifications is crucial for appreciating the complexities of life forms. So here we have types of organisms that I mentioned we will discuss. So we have two types of organisms: unicellular organism and multicellular organism. Unicellular organism, as you always comprise a single cell and have simpler metabolic processes and that produces rapidly. Multicellular organisms are comprised of multiple cells or individuals within organs and exhibit complex pathways, complex behaviors, and complex structures. Now each organism are made up of cells. So cells are the basic unit of any organism. So there are two types of cells. Basically, prokaryotic and eukaryotic are the two fundamental types of cells that makes up any living organism. They differ in their structure, organization, and complexities. So here, as you can see, I have listed out few differences in prokaryotic cells and eukaryotic cells with few similarities in them. Say, for example, having a plasma membrane, DNA, etc. And difference like prokaryotic would be unicellular, but eukaryotic may be unicellular and multicellular. Prokaryotic cells have sulfur DNA, but eukaryotic cells have linear DNA. So both differences and similarities have been discussed here, which would be discussed in details next classes. So in this unit, we will also be talking of compartmentalization of organisms. So here, compartmentalization of function to lower and higher organisms will be discussed in this unit. With involved understanding like how different cells or organisms contribute to the overall function of an organism, and we also touch upon like common physiological events that takes place, such as respiration, respiratory quotient, which gives insights into metabolic processes. So basically, compartmentalization refers to the organization of cellular functions into distinct compartments or organelles within the cell. So these compartments are separated by membranes that control movement of substances in and out, allowing for specialized function to occur within regions of the cell. So talking of compartmentalization in prokaryotes, so here, such as prokaryotes such as cyanobacteria, despite lacking of membrane-bound organelles, compartmentalization has been seen through enfolding of the cell membrane, which enables efficient resource utilization by concentrating enzymes and molecules for specific functions, including photosynthesis. So this organization also protects essential biochemical processes from harmful interaction and enhances energy efficiency by localizing reactions for optimal ATP synthesis. But in contrast, eukaryotic cells, which benefit from membrane-bound organelles like nucleus, mitochondria, are specializing in distinct functions such as genetic control, energy production, and photosynthesis. So basically, this compartmentalization facilitates the division of labor, communication among organelles, which enables vital communication, protection of delicate biochemical processes, and also fosters adaptability through evolution, and thereby contributing significantly to the efficiency and diversity of cellular forms, which we will be discussing in this unit. So next, we have subcellular organisms. So here we'll examine the structural functions and interrelationship of various subcellular organelles, such as nucleus, mitochondria, and plasmid and others. So what are the subcellular organisms? So subcellular organelles are specialized structures within a cell that perform distinct processes that are necessary for cell's life and function. Each organelle has its unique structure that suits its specific role, contributing to the overall functioning and efficiency of the cell. So the major cellular organelles might be nucleus, mitochondria, chloroplast plant cells, endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, ribosomes, cytoskeleton, plasma membrane, vacuoles, centrosome, and centrioles, as you can see in the figure, and these all will be discussed in this unit. So next, we have SI units in metric system. So in biochemistry, precise measurement is very crucial. So we cover the SI unit for mass, volume, temperature, lifetime, along with an overview of metric system in this unit. So the international system of units, that is SI, is modern form of metric system and is most widely used system of measurement worldwide. It is basically based on 7 fundamental units from which all other units are derived. These 7 fundamental units are meter for length, kilogram for mass, second for time, ampere for electric current, Kelvin for temperature, mole for substances, candela for units intensity. So these fundamental units can be also combined to form derived units such as joule for energy, Pascal for pressure, liter for volume. So in biochemistry, precise measurement are crucial for accuracy, reproducibility in experimental results, and these units also provide a standardized way to quantify and compare these measurements. The metric system, on the other hand, is a decimal-based system of measurement which is used internationally, and it is straightforward with the units that scale for the power of 10, making it easy to convert between different magnitudes. Say, for example, if length is measured in meter, that could be converted to millimeters, as in 10 raised to the -3 meters, and similarly to centimeters. So these all calculations about SI unit metric system we will be discussing in this unit. So next we have atomic and molecular concepts. So here we will be discussing key concepts of atomic weight, molecular weight, equivalent weight, as well as basicity and acidity of bases. So atomic weight is nothing but the atomic mass, which is the average mass of atoms of element that is calculated using relative abundance of isotopes in naturally occurring elements, and is usually expressed in Kelvin or atomic mass unit, that is AMU. Molecular weight also, as we know, is known as molecular mass and is sum of atomic weight of all atoms in the molecule. It is expressed in atomic mass units or diagrams. Now, for calculating this atomic weight of each atom in molecular formula, say for example in molecular water, since the formula is H2O, so we'll be adding 2 into atomic weight of hydrogen plus 1 into atomic weight of oxygen. So that gives, if atomic weight of hydrogen is 1.008, so 2 into 1.008 plus 1 into 16 would give you 18.0116 AMU. That is molecular weight of water. So this is how molecular weight is calculated. We will be teaching this calculations in detail in this unit. Next, we have equivalent weight. So equivalent weight is a mass of substance that combines with or displaces a fixed amount of another substance in a chemical reaction. Say for acids, it is a mass that supplies 1 mole of hydrogen ions for bases, 1 mole of hydrogen ions, and for bases, it is mass that supplies 1 mole of hydroxide. So likewise, the equivalent weight is calculated depending on the mass of substance in a fixed amount, however it displaces in a reaction. Now, basicity of acids would be the number of hydrogen ions that one molecule of acid can donate. Say, for example, in case of monoprotic acid, it can donate only 1 hydrogen ion per molecule, as in case of HCl. Likewise, we talk of acidity of bases, which refers to the number of hydroxide ions that one molecule of base can accept or neutralize. So it indicates the strength of bases in terms of ability to donate hydroxide ions in solution. So here in this unit, we will also be dealing with atomic and molecular concept, calculating these molecular weight, atomic weight, equivalent weight, basicity, and acidity of the bases. Further, we have Avogadro's number in molar calculation. So understanding Avogadro's number, molarity is essential for various biochemical calculations. Avogadro's number is fundamental constant in chemistry and represents number of constituent particles that are present in 1 mole of substance. So the value of Avogadro number is approximately 6.022 to 10 raised to 23, which indicates that 1 mole of any substance would contain exactly 6.022 to 10 raised to 23 particles. Next, we have mole concept. So here, mole concept is a method to use to quantify the substances in chemistry. It is based on Avogadro number and allows chemists to count the particles by weighing them. So 1 mole of substance is the amount that contains Avogadro number of particles. Avogadro number, as I mentioned, is very critical and constant in chemistry, providing a bridge between the atomic and microscopic worlds. It standardizes the measurement of chemical substances, allowing accurate molar mass determination, calculations, concentration preparation, and application of gas laws. So understanding these fundamental concepts is crucial for studying any chemical reactions, stoichiometry, or various applications in biochemistry and related fields. Atomic weight basically gives you insight into the relative masses of different elements. Molecular weight helps in quantifying different substances. Equivalent weight helps in understanding their reaction and stoichiometry. And the basicity and acidity of bases would help to predict the behavior of particular acid or bases in a chemical reaction, which is responsible for some sort of biochemical processes happening in our body. So this also we will have this in this unit. Next in this unit, we have, as mentioned, molarity, non-active molarity calculation. So molarity is nothing but the number of moles of solute per liter of solution, and it is expressed in moles per liter. The formula would be moles of solute divided by liters of solution. So, for example, if it is 1 mole of solution of sodium chloride, it would contain 1 mole of NaCl dissolved in 1 liter of solution. So normality, on the other hand, is number of gram equivalent of solute present in per liter solution. So it is expressed in equivalents per liter, and formula would be equivalent of solute that is divided by liters of the solution. Say, for example, for an acid-base reaction, where sulfuric acid is used, so the sulfuric acid, 1 normal sulfuric acid solution would contain 1 equivalent of sulfuric acid per liter, because sulfuric acid can donate 2 protons per molecule. So we can also say 1 equivalent is equal to half mole of sulfuric acid. Next, we have molarity, that is number of moles of solute per kilogram of solvent, and is expressed as moles per kilogram. So when we talk of formula for molarity, it could be moles of solute divided by kilograms of solvent. So if we say a 1 mole of solution of sodium chloride would contain 1 mole of NaCl dissolved in 1 kilogram of water. So likewise, we have calculations. Next, we have Dalton's atomic theory. So here, John Dalton, basically, who was an English chemist, formulated the atomic theory in 19th century, where he gave 3 concepts, that is, elements consist of atoms, and compounds. Where he meant that compounds are formed by combination of atoms of different elements in a fixed and simple whole number ratios. So chemical reactions involve rearrangements of these atoms, which are neither created nor destroyed in the process. Next, we talk of conservation of mass. So total mass of substance involved in chemical reaction remains constant and consistent with the law of conservation of mass during Dalton's theory. So also we have here the mole concept. So the mole concept is fundamental principle in chemistry or biochemistry that allows the biochemist or a chemist to count the particles by weighing them. It bridges the gap between atomic scale and microscopic scale, making it possible to quantify any substance in a practical way. So 1 mole of amount of substance would contain exactly Avogadro number of particles. Now molar mass is nothing but the mass of 1 mole of that substance, which is expressed in grams per mole. Next, we would be dealing in this unit with oxidation number, density, and specific gravity. So oxidation number of an atom to compound is measured of degree of oxidation of that atom. Oxidation number would be demonstrated in several forms. Say, for example, for monoprotic ion, the oxidation number would be equal to the charge of ion. Example, in case of sodium, if it is Na+, it indicates that oxidation number is 1. Density is defined as mass of substance per unit volume. And likewise, specific gravity is the ratio of density of substance to the density of reference substance which we are using. So oxidation number provides insights into redox reactions, electron transfer, enzyme activity, which is essential for understanding metabolic processes and energy production in cells. Density is crucial for biomolecular separations, concentration measurements, understanding cellular properties, aiding in experimental techniques. Specific gravity, on the other hand, is used in clinical diagnostic, reagent preparation, quality control, ensuring accuracy and reliability for biochemical analysis and experiment. So understanding these concepts in an application is fundamental in comprehending any chemical nature of biological processes and conducting precise and meaningful research. So, to summarize, in this unit, we will give you a foundational understanding of origin of life, the types of organisms, the chemical composition of living beings, the essential measurements and calculations in biochemistry. So, I'm very excited to guide you through this topic. If you have any questions, you can feel free to ask these in lectures while attending the classes. So, let's make this learning journey engaging and informative. Thank you.